The journey of life on Earth is often imagined as a neat, linear progression from simple organisms to complex beings like ourselves. The reality, uncovered through the lens of modern genetics and paleontology, is far more fascinating and circuitous. Evolution is not an engineer designing new parts from scratch, but a relentless tinkerer, constantly repurposing and modifying existing genetic blueprints. The story of how a fish's fin became a human hand, or how a sea creature's simple nerve cord evolved into a complex brain, is written not in the invention of new genes, but in the creative reuse of ancient ones. This process reveals a profound truth: nothing in biology begins where we think it does, and our own bodies are living archives of deep evolutionary history. Consider the monumental shift of life from water to land. The classic image is of a fish struggling onto shore, suddenly developing lungs to breathe air. The truth is far more elegant. Fish were breathing air long before they ever left the water, using primitive air sacs. These sacs, which in many fish became swim bladders for buoyancy, were simply modified over eons into the lungs of land animals. The genetic instructions for building these structures were already present, waiting to be deployed in a new context. This theme of repurposing is universal. What we recognize as a bird's wing did not begin as a wing, but as a limb adapted for other purposes, its function and form transformed over generations. Evolution works by co-opting the old to create the new. The secrets to these transformations are often locked within the earliest stages of life. Embryonic development is a powerful window into evolutionary change. Scientists discovered that some salamander species can develop into two entirely different adult forms based on their environment, one adapted for water, one for land, all from the same genetic starting point. This plasticity hints at how major evolutionary leaps can occur. A dramatic example is found in our own distant ancestry. The humble sea squirt, a stationary blob attached to ocean rocks, shares a crucial developmental stage with us, a free-swimming, tadpole-like larva with a primitive nerve cord. The monumental leap to vertebrates likely occurred when an ancient sea squirt-like creature failed to complete its development, retaining its juvenile, mobile form into adulthood. Our entire backbone-centric body plan may be the result of an ancient case of arrested development. The mechanism behind these changes lies not in the genes themselves, but in their regulation. Our DNA is like a vast library of construction manuals, but the critical elements are the switches that control when, where, and for how long these manuals are read. A tiny malfunction in a genetic switch, a single molecular misstep, can have colossal consequences, like the difference between healthy red blood cells and those that cause sickle cell anemia. On an evolutionary scale, changes in these switches are the primary drivers of bodily transformation. The difference between a human brain and a sea squirt's simple nerve net isn't necessarily in the core genetic tools for building nerve tissue, but in the complex switches that dictate how extensively and intricately those tools are used. Mutation provides the raw material for this tinkering. Far from creating sci-fi monsters, most mutations are subtle variations in the size, shape, or timing of development. When a mutation proves advantageous, natural selection can spread it through a population. Remarkably, the fundamental genetic toolkit for building animal bodies is shockingly similar across widely different species. The same clusters of genes that organize the head-to-tail body plan of a fruit fly are at work in frogs, mice, and humans. The genes that instruct the growth of a limb are virtually identical in creatures as diverse as whales, bats, and lizards. They simply produce different outcomes based on how they are regulated. A fish's fin and a human hand are built by variations on the same ancient genetic theme. This leads to a humbling realization about the human genome. It is not a perfectly optimized, pristine code. It is a dynamic, somewhat chaotic historical document. Our DNA is filled with genetic elements that copy themselves, jump to new locations, and sometimes lay dormant for millennia. We carry within us the fragments of ancient viruses and the echoes of genes that once served entirely different purposes in our aquatic ancestors. Our genome is not a static monument to our current form, but an active, messy workshop where the projects of deep time are still underway. Understanding this compels us to see the interconnectedness of all life. The same genetic principles that shaped a flower or a fish are actively shaping humanity. The history of life is a tale of endless assembly, where every new form is built from the cherished, modified, and repurposed parts of the past.